All right, are there any questions? Yeah.
My question is for animal or any species,
Uh-huh.
do we have just like one DNA or multiple pieces of DNA?
Oh, you mean one DNA or? I,
Because I understand that DNA is just like a very huge polymer, right?
right.
Yeah, polypeptide. So my question is, should it be like multiple of those? I mean, each of them going to function something, and I mean.
No, if you look at the human genome, for example, there are 3 billion of those letters, okay? 3 billion nucleotides. However, the way it is organized is it is organized into 23 chromosomes, all right? 23 pairs of chromosomes, all right? So you do the math, okay? So like 23 times 2, that's like 46 chromosomes, all right? So it is distributed among those different chromosomes. So, I mean, right now we're just looking at the structure of DNA, okay, what information it stores and so on, okay. We have not looked at the organization of DNA because the storage is very, very compact, okay. So you have something like it would be like a string that is like 12 miles long, right? Yeah, in fact, if I remember the numbers correctly from the book, a string that is 12 miles long, all right, you're trying to put it into a small ball, okay, in a compact way, yeah. So for the packaging, yeah, I mean, again, if it is a bacteria, there'll be just one chromosome, one circular DNA, double-stranded circular DNA. But if you're talking about the human genome, we have 23 pairs of chromosomes, you know.
So for the Genome project, they discovered the map, so it's need the map of all those 23, right?
Yeah, they found out all of them, like all the 3 billion. And I think for one of them, it might be for some person. For another one, it's like some genomes that are mixed up, you know. Like you can mix it up and then go and sequence it too, you know. So, all right, any other questions? So this is where we stopped last time, that saying that in this chapter, we were going to focus on the mechanisms by which DNA is replicated, possible errors which occur during DNA replication are corrected. Uh, I'm sorry, the errors during DNA replication and the mechanisms that are used to correct them. Right. So by the end of today's lecture, we would have finished all of this stuff. Right. Now, most genes are short stretches of DNA which encode a single protein. Right. Roughly speaking, you know, a gene is a stretch of DNA which codes for one protein. Right. However, not all of the DNA in a gene is used to encode the protein that it specifies. Right. Because there are regions, you know, that will specify whether a particular gene will be active or not, whether a particular protein is going to be produced or not. So those are called regulatory regions, right? They specify when and in what amounts the protein encoded by a gene is made, right? And the complete set of information in an organism's DNA is called the genome. So when you're talking about the human genome, means you need to know all the letters, those 3 billion letters that are there in human DNA. Okay, and they are different. Like they'll be a little different between me and you and so on. People who are related, all right, they will have similarities in the sequences, right? And you know, towards the end of this course, we will see how you can use this information to nail down the suspects in a criminal investigation, right? You hear about DNA technology. That guy has spent 30 years in jail, and now with DNA technology, right, he's been set free, okay, because he's not the murderer, right? So you can figure that out, you know. And I mean, you can calculate probabilities, all right? I mean, like the probabilities are staggering. You can say that there is a 1 in 5 billion chance or 1 in 6 billion chance that this guy committed the crime, okay? That means practically he did not, okay, because there are 6 billion people in this world, right? But that's where the lawyers get a chance to argue and all that. And when we get to that stage, I will tell you, you know, what the techniques are that are involved over there, right? So the genome is the complete set of information in an organism's DNA, right? If you're talking about the human genome, it's the 3 billion nucleotides that is there in a typical human cell, right? The Human Genome Project was the first attempt to sequence those 3 billion letters, okay, find out one by one, and it was a laborious process taking many, many years, right? And lots of money. Now, every time the cell divides, it is going to make a copy of itself, all right? So the two daughter cells will have to be identical. So at each cell division, the cell must copy its genome in order to pass it on to both the daughter cells. Now each strand of DNA contains a sequence of nucleotides which is exactly complementary to the nucleotide sequence of its partner strand. Right? I showed that to you last time. That A on one strand is going to pair with T on the complementary strand. G on one strand is going to pair with C on the complementary strand. Okay. So because of that, if you know the sequence on one of the strands, you automatically know the sequence on the other, right? And not only that, therefore each strand could act as a template or mold for the synthesis of the new complementary strand. So this is how DNA replication or DNA synthesis takes place, right? So you have two strands of DNA, one is S and one is S prime, right? And you can see the complementary base pairing. A pairs with T through 2 hydrogen bonds, G pairs with C through 3 hydrogen bonds, right? So in order for you to replicate this DNA, what is going to happen is that the two strands will have to come apart. Each one will serve as a template for the synthesis of the complementary strand. Okay. So what are the steps that are involved in DNA replication? First, the two strands have to separate out, right? So you now have single-stranded DNA. Each strand can then be used as a template for synthesizing its complementary strand. Then the newly synthesized strand and the template that is used to create it, they will pair together to form a new double helix, right? So based on this procedure, you can see that DNA replication is what is called semi-conservative, because the two strands come apart, right? Then this one serves as a template, right? But this template is now going to pair with the newly synthesized DNA, okay? So it is semi-conservative. At each step, you are going to get one strand from the previous step, okay? And so in each round of replication, one of the strands in each double helix is conserved from the previous round of replication. And all these things, biologists had to carry out experiments to see that, okay? They had to label the DNA, okay? Then let it come apart. Then when the new DNA is synthesized, okay, is the label still there? That's how they figured out, right? I mean, these are not automatic. Like, we are presenting it to you as though that's like gospel truth, but people had to do experiments to figure out whether that was indeed the case or not, and they had different hypotheses that were tested out. Now, if you're talking about DNA replication, that is going to start at what are called replication origins, where the two DNA strands will come apart, right? And the separation of the two DNA strands usually is carried out by initiator proteins, which will bind to the DNA and pull the two strands apart, right? Now, as I pointed out before, if you have AT base pairs, okay, that's 2 hydrogen bonds, right? If you have GC base pairs, that's 3 hydrogen bonds. So since DNA that is rich in AT, adenine and thymine base pairs, has fewer hydrogen bonds, it is usually easier to separate and therefore the site of replication origins. If you look at a bacterial genome, typically a bacterial genome will be in a circular chromosome, right? It will be one circular DNA molecule, right? So a bacterial genome usually has got only one replication origin because the genome of the bacterium is much smaller, right? All right? For the human, it's like 3 billion letters, right? And you're probably looking at like 20 or 30,000 genes. For a bacterium, it might be 3 or 4,000 genes, right? So it's much smaller. But in the case of the human genome, every time the cell divides, right? Every time the DNA is replicated, you have to make those 3 billion copies, right? You have to copy something that is 3 billion letters long. So that would take a long time. So you would want like some parallel processing to go on, right? And therefore you would probably have 10,000 replication origins, right? And simultaneously replication will start from each of these locations so that in parallel the entire DNA is getting replicated, okay? Different parts of the DNA. Now the core of the DNA replication machine is formed by an enzyme which is called DNA polymerase. Right? If you remember in the last chapter, we talked about proteins that synthesize polymers. They are called polymerases. Right? DNA polymerase is a good example of that. So this enzyme synthesizes the new DNA using one of the old strands as a template. Right? And so if you have this as a strand, okay, as a template strand, DNA polymerase is going to get on this and start making the complementary strand. That means T, G, I'm sorry, C, G, T, A, right? It'll create that strand, okay? So this enzyme catalyzes the addition of nucleotides to the 3' end. Remember, whenever you have a strand of DNA, there is a definite polarity. At one end, you have the 5' phosphate group, right? At one end. The other end, you have a 3' hydroxyl group on the sugar, right? And again, I mean, these are diagrams that I have covered. I don't have the figure here, right? But you can use your imagination or memory or whatever, okay? I'm assuming that you're studying because at the end of the day, what I'm lecturing here is a nice story to listen to, but you have to take a test, right? So just keep that in mind, right? So this enzyme is going to catalyze the addition of nucleotides to the 3' end of a growing DNA strand. So new nucleotides will come in here, catalyzed by that enzyme. And to form this DNA strand, there will be the complementary strand that this enzyme will look at and make sure that the base pairing is correct. Right? So it catalyzes the addition of nucleotides to the 3' end of a growing DNA strand by the formation of a phosphodiester bond. Right? We talked about that. Right? Phosphase, phosphoester bond, phosphodiester bond between this end and the 5' phosphate group of the incoming nucleotide. Phosphate group is schematically represented here by these yellow dots. Now, we know that DNA synthesis is not an energetically favorable reaction, right? So you have to drive it in a special way, right? Otherwise, it's never going to happen because it's an order-generating reaction. So how is that kind of a reaction accomplished? These nucleotides, they will enter the reaction initially as energy-rich nucleoside triphosphates. So you'll have the DNA base, all right, the 5-carbon sugar, that's deoxyribose, and then 3 phosphates, all right? So now I have these high energy phosphoanhydride bonds, right, between the first phosphate, the second one and third one, okay? So these can supply the energy that is needed, right? The free energy that is needed for this reaction to go forward, because otherwise this reaction will not occur because it's an energetically unfavorable order generating reaction. So the hydrolysis of one phosphoanhydride bond, right? So probably it's the second one from the sugar, all right? So it's this one in the middle, right? That one is going to get hydrolyzed. So then let's say you had ATP or GTP or CTP or TTP, that will now become just the monophosphate, okay? And that's the thing that can be linked, right? So the hydrolysis of one phosphoanhydride bond in the nucleoside triphosphate provides the energy for the condensation reaction, which will link the nucleotide monomer, all right, or the monophosphate to the chain, and it is going to release the pyrophosphate, the 2 phosphates that are linked together, right, into the environment. But then in the next step, even that phosphate is broken up, right? So you want to, so the cell basically wants to eliminate any possibility of that the DNA strand disintegrating, right? So the first step is to drive the reaction forward. You break down one of those phosphoanhydride bonds. You release pyrophosphate, right? The monophosphate has already been incorporated into the growing DNA chain. But then this pyrophosphate, the 2 phosphates that are linked together, even that phosphoanhydride bond, which is a high energy bond, is also broken, right? And so you have 2 inorganic phosphates that are released, and this makes the reaction virtually irreversible, right? Now there are certain complications associated with DNA replication, like there are some subtle details, okay? So in the top picture, right, you have just original double-stranded DNA. Now the replication the top strand is running from 5' in the left to 3' in the right. The bottom strand from 5' in the right to 3' in the left, okay? Now, in order to have DNA replication, you're going to need replication origins where this thing will open up, right? So initially it will open up at the center of this, right? It opened up, okay? But then as it is opening up more, so this is called the replication fork. It looks like it is moving in this direction, right? As the thing is opening up more, okay? So this picture shows you an opened up single-stranded DNA templates that are ready for DNA synthesis, right? Now the enzyme, so each of these guys will be used as a template. So the enzyme will get in here and it will start the DNA synthesis. This enzyme, the DNA polymerase, can synthesize DNA only in the 5' to 3' direction, right? So if you start from here, right, from the center of this replication origin, as the fork keeps on opening out, all right, this thing becomes longer and longer, and it's going to basically start making one strand of DNA like this, right? Okay. The problem is on this other half, because let's say it was open up to here. Initially it started making like this, all right, going in the 5' to 3' direction. As this one opens up more, it cannot go in the opposite direction, right? The DNA polymerase cannot go in the opposite direction, so you cannot synthesize that continuously. I have another picture that is going to show that more clearly, right? So, and that's what makes this a little bit more complicated. All right, are there any questions so far? So DNA polymerase can synthesize DNA only in the 5' to 3' direction, right? And this will cause a problem since the replication fork is asymmetrical, right? You have a replication origin and then it's opening out as a fork, all right? And I'm going to show you in the next figure that it is indeed asymmetrical, all right? And there is a problem here. The DNA strand whose 3' end has to grow as the fork moves is synthesized continuously, and this strand is called the leading strand and the other one is called the lagging strand, right? And let me illustrate that with a picture, right, on the next slide, right? So here you have the DNA template. The red ones are the templates, right? The DNA template. The top one is going from 5' on the left to 3' on the right. Bottom one going from 5', right, on the right to 3' on the left, right? Now, the replication origin will basically be at the center of this fork, right? So when this thing opens up and then it starts moving in either direction, everything is opening up, right? You can see that for the green one on the left hand side, the top green one, right, you don't have a problem. As this thing is opening up, this green one can keep on synthesizing DNA, right? DNA polymerase can keep on synthesizing DNA because it's still moving in the 5' to 3' direction, right? Same thing for this bottom one here, okay? As this thing opens up and moves more to the right, it can keep on, if it started here, as it's opening up, it's still synthesizing in the 5' to 3' direction. So it can be synthesized as one continuous stretch. But on these other two strands that are called the lagging strand, right? You can see that initially maybe it opened up somewhere here, right? So this has started synthesizing. But now this thing is opened up, right? So it cannot go from this here to there, right? It cannot go in the opposite direction. It can only synthesize in one direction, right? So there is a problem. So that means some other DNA polymerase will probably have to get in here and then synthesize this portion, right? So it'll have to be synthesized in bits and pieces, right? On the lagging strand. On the leading strand, you can do it in one shot because you do not have to synthesize in the opposite direction. You can go in the 5' to 3' direction, and as the replication fork opens up, right, as it keeps on growing, you are still continuing in the correct direction. So the synthesis, entire DNA synthesis on the leading strand can be carried out in one step. All right. On the lagging strand, it has to be carried out in different steps. All right. I mean, you first do it, then you cannot go back that way. So maybe another molecule of DNA polymerase has to get in and start a new strand. Right? And that's why you see these individual pieces here. And they have a name. They are called Okazaki fragments, named after the Japanese guy that first discovered them. Yeah. Hmm? No, synthesis is from the 5' end to the 3' end. Not on the template, but on the actual strand that is being synthesized. Yeah. Only one way, only one way. DNA polymerase can go only one way, yeah. Yeah, I'm getting to that. How to connect these pieces? Yeah, that's a good question, right? I'm getting to that. They will get connected, yeah. So, all right, any other questions so far? Now, the DNA polymerase, the reason it can synthesize only in one direction is because DNA polymerase is a self-correcting enzyme, right? It goes and checks its work, right? Like error correcting codes or whatever if you want to think of it. It has a proofreading activity or error correcting activity. And what it does is that before the enzyme adds a nucleotide to a growing DNA chain, right? It is going to check to see whether the previous nucleotide added has been correctly base paired to the template strand, right? So before they let you enroll for the PhD, they'll make sure that you cleared your master's, right, or your bachelor's, right? So it's doing nothing different, okay? But it goes and checks its work, okay? Is the previous thing correct? If the previous nucleotide has been correctly base paired to the template strand, then the polymerase will add the next one. If it is not, it's going to go and take the previous one out, right? So it's like, I mean, many times we get students who are not ready, if we send them back to high school again or something like that, So we don't do that, but I mean, it's something like that, right? It's going to go and take that out, and then it will try again. So thus, DNA polymerase possesses a 5' to 3' polymerization activity, where it is adding more nucleotides, okay, creating a polymer, and a 3' to 5' direction, it possesses a nuclease activity. Nuclease is something that disintegrates a nucleic acid, right? So in one direction it has polymerization activity, so it's a synthesizer. The other direction it's a degrader. Okay, so that's why it can synthesize only in one direction. Yeah.
Could you give one example of something like a mispaired nucleotide? I mean mispaired.
Oh, mispaired nucleotide, yeah.
So like AT, maybe AZ and AC.
Yes, yeah.
Oh, really?
Yeah. Yeah, instead of AT, it could be ACAG, whatever. Yeah. That would be mispaired, and it can happen. Believe it or not, anytime you step out in the sun, some of your cells have been messed up, all right? But we have repair mechanisms, so I mean that's why we are lucky. There are some people who don't, okay? It's very rare, but you can have diseases where, yeah, go ahead. Oh, nuclease means, see, okay, protease is something that breaks down a protein, right? Nuclease means it breaks up nucleic acids, right? See, nucleic acid is formed by linking nucleotides together, okay? So you need a polymerase to create a nucleic acid. But if you want to tear up if you want to chew up that nucleic acid, that means break it up into the individual nucleotides. Nuclease is nucleic acid degrading activity, okay? So because of the proofreading activity of DNA polymerase, a DNA polymerase cannot start a completely new DNA strand. See, one thing is because of the proofreading activity, it checks its work and it has got nuclease activity in one direction, right? So it can synthesize only in one direction. The other thing is that, if it is always going to go and check and see if the previous one is properly base paired, okay? Obviously, it cannot start a new DNA strand from scratch, right? Because if it tries to do that, if you're starting from a new DNA strand, where is the previous one to check, right? There's nothing to check. So it has the bad habit of going in, well, it's a good habit actually, but if you like to think of it, it has the bad habit of going and trying to check its previous work, but then how do you get started, right? Because when you're starting, there's no previous work to check, right? So that's the situation that we are in, right? So it cannot start a completely new DNA strand. So to start a new DNA strand, a different enzyme is needed to begin such a strand. That enzyme does not synthesize DNA, but it synthesizes an RNA strand. Right? RNA is ribonucleic acid, about 10 nucleotides long. Right? That strand will be about 10 nucleotides long, using the DNA strand as a template. And this RNA strand that it synthesizes is called a primer. Right? And the enzyme that synthesizes it is called a primase. Okay? So it will look at the DNA, right? Then the first 10 of them, because this primase is not going to check its work, right? It's going to just go and put in those nucleotides, right? And it can make more mistakes also than DNA replication, right? But it'll do that just to get started, okay? It's like jump starting a dead battery. Just to get it started, you have an RNA primer, right? And then now the DNA can latch onto it and do its work, right? DNA polymerase. All right, any questions? Now again, I mean, we go back to that picture, right, where we have the replication fork, right? That we have the replication origin in the center, then moving out in the form of a replication fork, and then you have these newly synthesized DNA marked in green. This is the leading strand. This is the leading strand. These are the lagging strands, okay, that are actually fragmented, right? So for the leading strand, you will just need an RNA primer only once, because once you start it here, it'll proceed continuously. You don't need any primers anymore, right? For the lagging strand, every time a new Okazaki fragment is produced, it's a new DNA strand that you're having to start. So you will need a primer here, need a primer here, here, okay? So now to get back to, was that your question, Hundo, or whose question about how we get rid of those fragments, right? So to produce a continuous new DNA strand from the many Okazaki fragments synthesized on the lagging strand, 3 additional enzymes are used, okay? So what should be done? I mean, in order to make a continuous DNA strand, right? First thing you'll have to do, right, is basically cut out that primer, which is really RNA, not DNA, okay? Then put in actually a DNA thing, right? Cut out the primer, put in the DNA, right? Replace it with DNA, and then there'll still be a gap that is left, okay? And then you have to stitch the gaps between them using another enzyme that is called DNA ligase, right? So everything is done by enzymes, right? So in order to produce a continuous DNA strand from these discontinuous fragments, right? You need 3 things. You need a nuclease, right, which disintegrates the RNA primer, right? Again, nuclease means something that degrades or disintegrates nucleic acids. Then you will need a repair polymerase, which will replace the disintegrated RNA with DNA, right? Because you're trying to replicate DNA, not replicate RNA. And then you also need what is called DNA ligase, right? So it's something that stitches the 2 fragments together, which will join the 5' phosphate end of one DNA fragment to the 3' hydroxyl end of the next one, right? And that's how you will get continuous DNA strand, right? Now, if you look at the book, the reference book that I've given, I mean, I don't have the latest edition, and somebody, picked up my old edition. I don't even have that either. Okay, so it's gone, right? I acquired it in 2002, 2003, so some student disappeared with that. That's okay, 10 years I can probably get another one, okay? But I'm saying if you look at that book, right, even the newer edition, they will, try to give you a schematic diagram of how probably everything works together. Because remember, this thing is being discontinuously synthesized. That's being continually synthesized, okay? But if you look at it, everything is happening synchronously, and everything seems to work fine. So it could be part of a big molecular machine, okay, that is carrying out this part discontinuously, that part continuously, cutting out stuff here, replacing it, and then you get the continuous DNA strand at the end. Now, what are the proteins that are used in replication, right? There are several proteins that are needed, and these proteins will be present in each and every cell that divides, because that will have to replicate its DNA. So the first protein is the one that we already discussed, that is DNA polymerase, okay? Then there is another protein that is called primase, right? That's the one that basically produces that RNA primer. We have discussed that too. Then you have helicase, which is a protein which uses the energy of ATP hydrolysis to move along the DNA, opening the double helix as it moves, right? See, for any cellular activity, if there is movement and all that, you're going to need energy, right? And if you need energy, that has to come from the hydrolysis of ATP. Then there is another protein that is called the single strand binding protein, because when the two strands separate out, okay, they will have a natural tendency to again form base pairs and form the double helix. So once you separate them out, you have to hold them in place. So there is some protein that does that. That is called the single strand binding protein, which will cling to the stranded DNA exposed by the helicase and prevent it from immediately forming base pairs. Right? Then there is also a sliding clamp, which will make sure that the DNA polymerase is firmly positioned on the DNA template as the polymerase moves along. And of course, on the lagging strand, because you have these Okazaki fragments, so you have to let go of the polymerase, then let it jump to a new location. So the sliding clamp will have to release the polymerase every now and then as necessary. Now, for us, that detailed machinery is probably not that important. You need to know about the Okazaki fragments, about the discontinuous synthesis and all that, but exactly how all the clamp and everything works together, it's probably not that important for us. And some of it may even be speculative. At least it was like 10, 12 years ago, but maybe not anymore. Okay? Because people think about, that it's possibly happening that way. They postulate models, but those may not have even been validated. But what is important for us is to know that because all of the information that specifies the living organism is contained in its DNA, cells will go to great lengths to maintain the integrity of the DNA. Right? You do not want changes in the DNA. Right? If your DNA changes too fast, then you're in trouble. Right? You're not going to be able to function properly. Right? So one aspect of maintaining this integrity is that DNA replication must be carried out very, very accurately. In fact, the proofreading activity of DNA polymerase, this tendency of DNA polymerase to go and check the previous nucleotide, right, for correct base pairing, this tendency ensures that in DNA replication, only about one error is made for every 10 to the power of 7, right, nucleotides copied. Okay, so every 10 million nucleotides copied, you make one mistake. Right? And that's pretty good. In fact, that biology book, has a table. These are the number of mistakes on average the United States Postal Service makes, right, in deliveries. FedEx makes, and this one beats it, by a lot, okay, by orders of magnitude, right? But that's not enough. It's even more accurate than that. In addition to, so the possibility of making mistakes is 1 in every 10 million, okay? But you're making 3 billion letters you're copying. Every time the cell is dividing, the DNA is being replicated. So there are repair mechanisms. Let's say despite this low error rate, there are still mistakes that are being made. So there are DNA repair mechanisms that will ensure that this accuracy is enhanced even further by a factor of 100. So that in the end, right, in DNA replication, there is only one mistake made for every billion nucleotides copied. Now, so you have 3 billion letters, you're making one mistake every billion nucleotides copied, okay? And you have all these repair mechanisms. If there is a mistake, if the cell detects that there is a mistake in the DNA, and again, I mean, you have 2 copies of the information. So chances are, if on one copy something is messed up, on the other copy it's fine, okay? So it can use the other copy as a template and fix this, all right? There could be exceptions. If somebody gives you such a high dose of radiation that both your strands of DNA are damaged, then you don't have anything to copy from, and that can happen in outer space or something like that, if you're going there. So despite the elaborate repair mechanisms that are in place, sometimes there'll be a permanent change in the DNA, which is called a mutation, can occur. Such changes over the long run, billions of years, have led to the wide variety of living species that we see on Earth today. If you start from a bacterium, right, how is that going to evolve? Under selective pressure, there will be mutations, right? And it's going to try to survive, right? Like if conditions on the Earth changed, okay, like it became really hot, most of us will be dead, right? But maybe 1 or 2 of us will evolve, right? We can tolerate high temperatures and we'll be okay, right? And we'll take over. So that's how, evolution has taken place over time, right? However, so this business of mutating your DNA, all right, to confer survivability in a changing environment, that's a good thing, right? So for individual cells, that helps them. But if you're looking at multicellular organisms like us, all right, if you have this kind of mutation, then that's disaster, okay? Because it's not good. You, if somebody gets cancer, the cancer cell in your body is having a ball, okay? They're having a good time, they're surviving and all that, but they kill you, right? So in the short run, they can spell disaster for the organism. For example, cancer is a disease that can result from the accumulation of DNA mutations. And some people are more prone to it because possibly from your parents you inherited a gene, right? Some DNA that already has a defect, right? You have 2 copies of each gene. So if one is already defective, right? If somehow, so you just have one more to go, right? If that other one gets mutated, then you could get full-blown cancer. Right? And there are people that get that. Now I want to introduce some terminology that is related to genetics and heredity. If you try to read any paper in the genomics area, even biology or bioinformatics, right, you'll come across this terminology. So what is a gene? A gene is generally a stretch of DNA which codes for a protein. Then there is this terminology that is called an allele, right? An allele is a variant of a gene, right? See, although different individuals from the same species, they may possess the same gene. Like you have a hemoglobin gene, I have a hemoglobin gene, right? But the exact DNA sequence will not be the same. It will be a little bit different. So we have different alleles of the same gene, right? Now again, I mean, there is this terminology called wild type allele. And this is real biological jargon, right? Just like in, in electrical engineering, we talk about time constants and things like that, Q factor, and this is what the biologists will talk about. So if you don't know this terminology, you will have no clue about what they're discussing. Yeah, go ahead.
How can you realize if it's an allele or it's a gene? I mean, where's the...
No, allele is a variant of a gene.
Yeah.
Okay, so
I guess that there
no,
are
no,
many...
there will be many alleles of the same gene, many alleles.
That I understand, but the thing is, what I'm asking is, at what point an allele is no longer an allele and becomes a different gene from its...
A different allele, you mean?
A different gene, like it's
Uh,
so different from the original...
no. It may lose function, but they will still call it an allele. Okay, they will call it a mutant allele. See, an allele is a variant of a gene. Okay, it may be a functional variant, right? It might be a dysfunctional variant. So if it's a functional variant, they'll call it a wild type allele. Okay, that is the type of allele that is usually present normally in the population. If it is something that is not the wild type, right? That's called a mutant allele. It is still an allele, right?
But still, my question is, how do you know two sequences or two sequences?
You don't know that unless there is, see, there could be sequences, right, where there is some stuff that is messed up. Even then you would probably determine based on loss of function. Okay, loss of function, not so much from the sequence. Like for example, if you're looking at diabetic mice, right, versus normal mice, the diabetic mice will not be able to produce insulin. Okay, it'll have the gene for insulin knocked out, right? So that's where it'll be a different allele. But other than that, Do you know a priori how many things you can change before it becomes dysfunctional? That's hard to say. In some cases, changing one nucleotide, right, among thousands or even millions is enough to cause loss of function.
Okay, so
Ano-
alleles may differ in one or two nucleotides,
or many more, many more,
or many more.
many more also, many more because
Here's my question.
yeah, yeah.
How many nucleotides you have to change?
No, 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 no. There is no formula like that because even in the human genome, right, there are 3 billion nucleotides, I told you, right? Only about 2% of that actually codes for anything, right? The rest of it is what they call junk DNA. They do not know. It may have evolved over time. Maybe thousands of years ago it had some function. We don't know the function. So which means that if you go and make some changes over there, okay, as long as you don't change the crucial parts, nothing will happen, right? It'll still be, it's a different allele because it's looking different. It's not the same sequence, right? However, but it's still probably a wild type allele because, it's functional, right? But if there are lots of changes or changes in a crucial region where you cannot produce that particular protein, right? That's when you'll have loss of function. But there's no way to predict that. Yeah, go ahead.
Does a gene always have the same position in the genome, for example? That's how they
Uh,
typically.
it Most of the time it probably will because certain genes are carried on certain chromosomes, all right? The way it is organized, all right? It is, but it can move around, okay? And I will discuss later on because it might get cut from here and it might be moved to some other region, all right? And that can cause problems, yeah.
Okay. So if we look at the chromosome in this part, for example, and look at another chromosome from another human being, for example, in this part, and we found that they're different, this means that these are two different alleles, or
No, if it is the same gene, okay, you would compare the same gene, right, to find the allele, all right? Chromosome is about how it's organized inside the cell, all right? And a lot of things can happen because, uh, parts of this thing can be, you know, cut off from the chromosome, then it can move around, go somewhere else. There is also examples of gene duplication. Like instead of having one copy, you have multiple copies of the gene, right? An example is being the globin gene for hemoglobin. We have 5 copies of the gene, right? One copy is utilized when you're an infant, okay? Another one as you go older and so on, okay? So there are different copies and, it has, because, ideally you want DNA not to change, okay? But it is true that, over evolutionary time things change, okay? You respond to the pressures in the environment. Anytime you catch a common cold, all right? Some of your stuff is altered, right? Because your cells have been attacked and the virus has put in some of its DNA, okay? So your DNA is not the same anymore, right? So you like it or not, that's the way it is, all right? So hopefully it hasn't altered something in a major way, right? That you die or something like that, but it will make changes.
how do you know that this is a gene and this is not just, how do you label this sequence as a gene and that sequence is not a gene and so on? Is it according to the product?
According to the product, yes, according to the product. That's a big good question, okay, and that's a question that many of you should focus on, because the 3 billion nucleotides that has been figured out, okay, but then what about the functions? Okay, that's not figured out as yet. Okay, there's functional genomics, all right, piecing together function, which gene controls which one, right? Those things are not well understood. Bits and pieces are, but most of it is uncharted territory.
So you basically use, you have like for certain proteins, which sequence it's, so which, for X sequences, which proteins
Yeah, yeah.
they produce, and that's the way you identify
hmm
different genes.
Yeah, that's how you identify different
That was
genes.
my question.
Yeah. No, which protein you produce, yes, yeah, which protein. And not only that, let's say if you know the hemoglobin gene in the case of humans, okay, let's say you've sequenced it, okay, you know what it is like. Chances are that in dogs, cats and mice, because we are pretty closely related, you can use the hemoglobin gene, all right, and use that as a probe. There will not be a perfect match, but you'll pick that up, right? So that's how it's typically done. Right? Actually, this is getting to your question. See, in many situations, altering parts of the genome of an organism will not result in a change at the macroscopic observational level, right? What protein is produced. Such a change is called a genotypic change or characteristic, where you just made some changes in the gene. You will not see any, effect outside, right? The person is still normal. On the other hand, a characteristic which manifests itself at the observational level is called a phenotypic change or characteristic. For example, you got a cancer or a tumor, right? Everybody can see that. Okay, you don't need to go and do DNA analysis to figure it out. You know? Now, in humans and many other organisms, each cell except the reproductive ones, they contain 2 copies of each gene, right? So there is some redundancy built in over there. Consequently, in such organisms, if you go and mutate 1 copy of a gene, you mess up 1 copy, you may not see any manifestation of the associated phenotype, right? Like let's say for the gene for insulin, if one of the genes is messed up, okay, you're not going to become diabetic, right? Or if of the genes for sickle cell anemia, right? Like hemoglobin gene, if you have only one of the copies messed up, it's not producing normal hemoglobin, you probably feel tired, right? A lot more than ordinary people, but you know you'll probably still be okay. In fact, I used to have a student that told me that he had he was a carrier of sickle cell anemia, so one of the copies was mutated. So he used to feel tired every now and then. Okay, I mean he volunteered that information. Otherwise, I cannot ask him for that. Okay, under the laws here, I cannot do that. Okay, I'll get into trouble. Right? But anyway, so a mutation where you need both the copies of the gene to be dysfunctional in order to see the effect, that is called a recessive mutation. Right? On the other hand, a dominant mutation is one where mutating 1 copy of a gene is sufficient for the phenotype to manifest itself. Right? Like if you mess up 1 copy, it's something like the dominant mutation is like the accelerator in a car. You need 1 accelerator, you press on it, the car will go. Okay. Whereas the recessive mutation, you can think of it as the brakes. So right, in a car, you have the handbrake and the foot brake. Okay, if one of them is active, unless both of them fail, right, you're not in trouble. Right. And even in cancer, the same kind of things happen. You know, there are brakes on cell division and there are accelerators on cell division. Accelerator, if one of them misbehaves, you're in trouble deep time. Brakes, one of them misbehaves, I mean, you're okay. Until both of them fail, you're fine. Now, when the 2 alleles for a particular gene, right, in a genome are of the same type, let's say the hemoglobin gene, you have 2 copies, both of them are wild type, then you say that the genome is homozygous for that gene. Right? Again, some terminology. On the other hand, when the 2 alleles are of different types, then you say that the genome is heterozygous for that particular gene. Okay. Now, DNA mutations can result in a lot of different diseases, right? And a good example is what is called sickle cell anemia, where your hemoglobin, right, is not the right shape. And so the cell has the shape of a sickle. It's not good enough for picking up oxygen and transporting to tissues, right? So this is an inherited disease which is caused by a single DNA-based mutation in the beta globin gene. The beta globin gene has got 146 amino acids in it, okay? Just 1 letter, right, in the code for that, among these 146 amino acids, each amino acid is coded by 3 letters, okay? So just 1 out of 146 times 3 letters, all right, messed up, that'll cause catastrophic loss of function. Humans have 2 copies of this gene, 1 inherited from each parent. To have full-blown sickle cell anemia, you must have 2 defective copies of this gene. So both your parents have to give them, you know, their bad genes, all right? And then you're in trouble, and then that can become a life-threatening disease, right? If you get only 1 bad gene from one parent and a good gene from the other, right? If both are carriers, you're still okay. You can function. So if an individual has got 1 defective copy, then he or she does not show symptoms of the disease, although they can pass it on to their offspring with another person having a defective copy of the same gene, right? Because there is a 1 in 4, if both the parents are carriers of sickle cell anemia, there is a 1 in 4 probability that the child will develop full-blown sickle cell anemia. Right?
Because the ones that are passed to the children are like random.
Yes, yes. We will talk about that later. Yes, they are at random. So you know, yeah. So hence a person with 1 defective beta globin gene is a carrier of sickle cell anemia. And clearly the mutation that is involved in sickle cell anemia, that is a recessive one, because both the copies have to be mutated before you get the disease, right? And the carriers of this disease, they'll have 1 normal beta globin gene and 1 abnormal one. So they have a genome which is heterozygous for the beta globin gene. Again, I'm just introducing some terminology. It'll help you when you read papers later on. Another disease that can be the result of DNA mutations is colon cancer. This is caused by the failure to repair DNA damage in the colonic cells, in the cells lying in the colon. All right. Believe it or not, the cells on your skin are replaced once every 10 days. The cells in your colon are replaced once every 3 days. Okay. It's a dynamic equilibrium. All right. And you have the ability to repair those cells. Now what happens if somehow that DNA repair gene, you didn't get it properly, right? It's not repaired. So the DNA is getting mutated more and more. At some point in time, there could be genes that turn on cell division, okay? That will be activated because of these mutations, and that's when tumors start to develop, right? So humans inherit 2 copies of a DNA repair gene, one from each parent. If one defective copy is inherited, then in that case you just have one more to go, right? So at some point in time the other one will also get mutated and you'll get colon cancer. And that's why this disease actually runs in families, right? I know somebody that was 1 year junior to me in college. He died of colon cancer, I think probably 15, actually more, yeah, maybe 15 years ago, right? 15 or more. And many years ago, I remember when I was in college, we heard suddenly his father had died of colon cancer, same thing. And then once he died of colon cancer, that time they screened his older brother and they found that he had polyps in his colon. So his life was saved, the brother's life was saved, but this thing runs in families. Now the incidence of colon and other types of cancers goes up dramatically with age, since unfortunately the DNA repair mechanisms slow down as one ages. Okay? It is not just that you look old, right? But the repair mechanisms also are slowing down. And also enough mutations have to accumulate before cancer can result. And so time is required for the initiation and progression of the disease. That's why cancer is typically a disease of old age. Okay? The older you are, the more your chances of developing cancer. Now without DNA mismatch repair, the mutations would develop much faster and the incidences of cancer would go up catastrophically. Right? The DNA mismatch repair system recognizes the errors in DNA replication and it will correct them and thereby prevents them from propagating. It is not yet known how the DNA repair machine preferentially acts on the newly synthesized DNA strand and leaves the original strand unchanged. Well, maybe it's known right now. My notes are like 10 years old, 10, 12, but maybe some guy doing like Nobel Prize kind of stuff might have found that out. I don't know. Okay. Now, in addition to errors during DNA replication, the DNA in our cells is continually suffering damage, either spontaneously or due to agents such as sunlight, chemicals, etc. Examples are depurination and deamination. Depurination means you lost a purine, right? Adenine and guanine, A and G are purines, right? Deamination means you lost an amino group. And another example is ultraviolet radiation in sunlight. So let me show you what happens. In depurination, what happens is that you have A and G are purines. Okay, so you have a guanine here connected to the 5-carbon sugar, deoxyribose, and then phosphate. In depurination, for some reason, this thing just left. Okay, so it's like a person that has got missing teeth. Okay, so now once this is left, if this strand is used as a template for DNA synthesis, mistakes are going to invariably occur because there's nothing in there, right? So this is depurination, and apparently this happens spontaneously because that biology book has some numbers that by the time you finish reading this page, okay, I don't know, several thousand of these guys are gone in your body, okay? They're gone. So, but it gets fixed, so we don't need to lose sleep over that, so, all right? So when such a strand with missing bases is used as a template, replication errors are bound to occur. Then in another example, in deamination, an amino group gets removed. So you have cytosine here, you remove the amino group, all right? Through the addition of a water molecule, it's gone. Now this thing, instead of being cytosine, has become uracil, right? So one DNA base has been transformed to another through the loss of an amino group. Now the problem is that C pairs with G, right? U pairs with A. So if you use this strand for synthesizing new DNA, you're going to get base substitution, right? Instead of what you were supposed to have, you'll have something else. Is that clear to everybody? And now comes the fun part. If you're looking at, I'm going to stop in a minute. If you're looking at stepping out into the sun, all right. So try to avoid the sun when you go out right now, okay? Because these are the thymines, all right, over here, 2 T's, all right. Because of the ultraviolet radiation in sunlight, these guys get linked together, right? See, and it produces a thymine dimer, okay. So that is going to confuse the DNA replication machinery. This needs to be cut out, right? So we have the right enzymes to do that. If it detects that this has happened, it'll cut that portion out, use the complementary strand, and produce the copy. Fortunately, we have that copy. We have the enzymes to do that. There are people who don't have that. Okay, if you're born without that, then you have a disease that is called xeroderma pigmentosum. You step out into the sun, you will get sunburn and blisters, right? And kids who have that disease, they cannot go out into the sun. The only time they can play is at night, okay, when you have floodlights, right? Because if you go into the sun, you're going to get skin cancer right away, right? So that disease is very much there. Okay, it's a rare disease, but it's there. Let's stop here for today, right? We're running out of time. But I hope you're finding some of this interesting, okay? And, in this course, you will pick up some, several interesting items, like you will understand how they track down suspects in a criminal investigation, okay? What is the exact technology used and so on, okay?